turning point?
No.
Do you have like any questions that you're going to be asking?
No.
Okay. All right.
Okay, thanks.
Check the battery on that as well. Sorry about that.
May not be real inside the entire.
Okay, let's get going. So I'm Dr. Newman. I'm going to talk about the extracellular matrix today and continue tomorrow with connective tissues and the day after with bone and cartilage. So what I want to do here is make a connection between what Dr. Luria told you about and what the next group of lectures is going to be. So you know about epithelial cells, and you know that they sit on a basement membrane. So the basement membrane is an interface between the epithelium and the underlying connective tissue. Connective tissue cells are not directly attached to each other the way epithelial cells are. They don't create barriers, but they fill space. So the first extracellular matrix that you encounter here is the basement membrane. And the basement membrane is complex. It's made of several layers. The part that is closest to the epithelial cells and depends on molecules that are produced by the epithelial cells is called the basal lamina. And the thicker layer beneath is called the reticular lamina. And I'm going to tell you about what they're made of and how they're organized and so on. Then I'm going to talk about what's called the interstitial matrix that is between connective tissue cells. So let's continue here. Now, in the lab, you're going to see images like this very often. And you see cell nuclei embedded in a material that looks pretty featureless. So for a long time, it was thought that there wasn't too much interesting going on there. And I'm going to try to persuade you otherwise. This is a highly structured, very kind of densely populated with molecules medium that is a repository of growth factors and mechanical signals and things that influence as the cells produce them in very specific ways. So just to kind of orient you here, all of these kind of dark oval structures are cell nuclei, and just beyond the cell nuclei you can make out in most of these the cytoplasm, but that's where the cell ends, and everything outside it is extracellular matrix. And extracellular matrix is mainly water. Even when the extracellular matrix is solid, the way it is in bone and tooth, the major component is water. But the water is structured by molecules called collagens, by molecules called proteoglycans, glycosaminoglycans, glycoproteins, and these are organized in very complex architectures. Okay, so if we look here, this schematic allows us to see the two main kinds of extracellular matrix. So here is an epithelium, and the epithelium has integrins at its basal surface, and the integrins are involved in hemidesmosomes and in complexes called focal adhesion complexes. I'll tell you about those a little bit later, but they are attached directly to the basal lamina and the basal lamina is underlain by the reticular lamina. So let's look over here. This little circle here is showing you the basal lamina, and this is a blood vessel, and the blood vessel is made up of an epithelial type of cell called endothelium, as you heard, and the endothelium is sitting on a basement membrane, which is now outside the blood vessel. Okay, so the apical surface of the endothelium is facing the inside of the blood vessel. Okay, so the little circle here is around also a basal lamina. Okay, and then we're looking over here, and we see molecules organized in a very highly structured fashion. Proteoglycans, type 4 collagen, laminin, and these have a particular arrangement within this structure. So type 4 collagen is a product of the epithelial cells, and it is the collagen that is closest to the epithelial cell layer. There is, as we move out, as you'll see later, the reticular lamina, the thick part of the basement membrane, is made up mainly of type 3 collagen. That's not represented here. Laminin is a protein that binds directly to the integrins of the basal surface and mediates attachment to the type 4 collagen. And proteoglycans are also involved. Proteoglycans are hybrid molecules, contain a protein, but a lot of polysaccharide chains covalently attached to them, sticking out of them like a brush. And those polysaccharides are called glycosaminoglycans. And they have that special name because most polysaccharides don't have sugars that contain nitrogen. Most sugars like glucose and fructose don't contain nitrogen. But the sugars that make up the glycosaminoglycans, which are the extracellular matrix polysaccharides, mainly contain nitrogens. They're glucosamine, galactosamine, so on. Okay, so that is the basal lamina. Now, if we look over here, we're outside of these epithelial regions, and these cells are connective tissue cells. They're called fibroblasts. And there are other kinds of connective tissue cells as well, but the major kind is called fibroblasts. And the major protein that they make is called type 1 collagen. There are 27 different types of collagen, and there are many more genes than 27 because some of the collagens are made up of several gene products. But in any case, the major protein of the body, the human body and the bodies of all animals, is type 1 collagen. So 25% of the protein in our bodies is type 1 collagen. Okay, so this is a less organized matrix. This is almost organized in an almost crystalline fashion. But here we also have proteoglycans. We have collagen. These triple helical structures are type 1 collagen. And we have a protein called fibronectin, which is a glycoprotein, and several other things. But water is very prominent here as it is here. Okay. So let's look at some of the molecules involved. And actually, on this figure, the molecules are drawn to scale. So this is fibronectin. It's a dimeric molecule. It has 2 chains, and they're attached by disulfide bridges at the end. This is a fibrillar collagen. Type 1 collagen makes fibrils. Type 2 collagen, which is very prominent in cartilage, makes fibrils. Type 3 collagen makes fibrils. So I could go through the whole 27 collagens, but in fact, type 4 collagen doesn't make fibrils. Type 4 collagen makes a kind of a lattice. It's, if we look back here, type 4 collagen, it's not organized into fibrils the way the type 1 collagen is. Okay. And everything green on this diagram is protein. Tenacin, laminin. Laminin I told you about. It's shaped like a cross. It has 3 polypeptide chains organized this way. Tenacin is a hexamer. And it's involved in most of the extracellular matrix glycoproteins are involved in adhesion, but tenacin actually interferes with adhesion. It kind of modulates adhesive interactions and blocks them. Then we come over here and we're looking at molecules that are entirely or partly polysaccharides, glycosaminoglycans. Hyaluronan is the longest of these, and it's the most widely distributed. And it doesn't have a protein attached to it. It's just a glycosaminoglycan chain. Decarin, perlecan, aggrecan, these are proteoglycans that are found in different tissues. And what you'll find is that the glycosaminoglycans that are associated with these proteoglycans are often identified by the tissue that they were first discovered in, although later on it was found that every one of them is more widely distributed. So for example, heparan sulfate, excuse me, the prefix refers to liver, hepatic, and that was first discovered in liver. Chondroitin sulfate was first discovered in cartilage. That prefix refers to cartilage. Dermatan sulfate was first discovered in skin, and similarly, it's more widely distributed than that. Okay, so here's an extracellular matrix that it's outside the cells. These are portions of cells here, but we're looking at type 1 collagen fibers, very densely packed. And this is a higher magnification here. And you can see that these fibers look like they have stripes on them. And this is because the way the fibers are assembled and organized is very regular. And when the electron micrograph is prepared and the stain is deposited, it deposits in a periodic fashion along these fibers and the fibrils that make up the fibers. So it looks like this is diagnostic of collagen fibrils and fibers, that they look striped. So how does that happen? So you have these collagen molecules, and I said that some of them are more than one gene product. Type 1 collagen is 2 gene products. Let's just say there's a beige gene product and there's 2 brown gene products. Okay, and they make a little braid, a little triple helix, and these molecules, which are outside the cell, then assemble side by side and end to end, so that they form these fibrils. Fibrils are the first level of organization after the molecule. And you can see each of these fibrils has this kind of structure. It's an organization of the collagen molecules. Okay, and then the fibrils also assemble side by side and to end, and they make fibers, which are the large structures that you saw under the electron microscope. And the fibrils and the fibers both appear striped. And in some tissues, it's mainly fibrils, but mostly it's fibers. And the fibers can organize in this kind of random meshwork, and then you have something called an irregular um, collagen matrix. But they can also line up again at a higher level of organization so that they're all oriented the same way. And then you have a regular collagen matrix. But that you have that in tendon and in ligaments. And I'll get into that more tomorrow. Okay, so that's the organization of collagen. Now, this is very unusual, and putting this here to show you that the physical organization, not just the molecular composition of the matrix, is very important. So this is the collagen of the cornea of the eye. And these are fibrils that are organized in a very regular fashion. And you go to the next layer, and they're also organized in a regular fashion, but here we're looking at cross sections. And here again, it changes back to that orientation. And because of this almost crystalline organization, it's called paracrystalline, light can pass through this matrix. Now, if you look at the dermis of the skin, it's made of the same kind of collagen, but light won't pass through it. It's completely opaque. So a good analogy would be carbon. Coal is carbon, and so is diamond. Light passes through the diamond, but it doesn't pass through the coal. So the organization, the crystalline structure, is important in the case of these biological materials for the function of the material. So you have to pay attention to organization as well as composition. Okay, so this is what happens when collagen molecules get made. They get made as peptides, and we're looking at type 1 collagen here. And these are called procollagen chains, and they are helical, but they have non-helical regions as well. And they have what are called propeptides at the C-terminal end and at the N-terminal end. And these, these peptides get nucleated by the propeptide, and then that allows them to assemble into the triple helix that I showed you before. And then this gets cleaved by a proteinase, and the N-terminal propeptides also get cleaved by the proteinase, and you wind up with a collagen molecule that allows you to assemble into fibrils. Okay, so a couple of important things here. You see these fibrils, they assemble in a non-covalent fashion, but then there are links between them, cross-links, and these cross-links are because some of the amino acids, lysine and proline, which are prominent in collagen, can make chemical structures called Schiff bases, and there is a non-enzymatic linkage that occurs between them. Okay, but in order for that to happen, enzymes have to put hydroxyl groups onto the amino acids. So you have hydroxyproline, for example, and that allows this linkage to occur. So the stability of collagen is very dependent on these linkages. If the metabolic steps, the biosynthetic steps that contribute to this cross-linking are impaired, you can have defective collagen and weak tendons and weak skin and so on, weak bones. So there are all sorts of genetic components to collagen assembly and function beyond the specifying the polypeptides themselves. Okay, another thing that's important here that I'll return to later is these proteinases. Excuse me one second. Most proteinases of the extracellular matrix depend on metals, tin, zinc, to function, and they're called metalloproteinases. and they're very important in a kind of equilibrating the structure, because some of them actually break down the matrix after it's assembled. And we'll get into that. Okay, so here is a chart with some of the collagens, type 1 collagen, type 2, where they are located and what their molecular substructure is. Okay, and this is, you can see that some of them, type 2 is the major collagen and cartilage. Type 3 is in reticular, the reticular lamina and reticular connective tissue, and they all have different gene products. Okay, and they have names like this. And this is an old chart. I stopped at 12, but we know there are 27 now. So this is what you need to memorize for the exam, plus all these references too. Just kidding. Okay, just gives you the idea that there are lots of collagens. Some are major collagens. Some are considered minor collagens. The minor collagens are minor because they're not very abundant, but they also play very important roles in the tissue. Okay. So let's now look at the collagens that are associated with, well, let's look at type 3 collagen. So I mentioned that underneath the epithelial cell, you have the basal lamina. I think Dr. Luria showed you this picture. It's organized. It looks kind of lightly staining and more densely staining under the EM. It's probably not true structural differentiation, but it just separates out like that. But anyway, there is the basal lamina portion, and then there is the reticular lamina. And the reticular lamina very prominently has type 3 collagen fibers. They're called reticular fibers. And the type 6 is anchoring fibrils. And there are anchoring plaques that are made of type 4 collagen. Anyway, it's a complex structure, but type 3 is the most prominent collagen in the thick region of the basement membrane. Now, type 3 collagen appears as the main matrix component of a certain class of connective tissues, which are called reticular connective tissues. So reticular connective tissues are found in the spleen, in the lymph node, the bone marrow, and the matrix fibers provide a framework for the attachment of lymphocytes and other cells. So these cells don't intrinsically bind to each other, so they need some scaffold in order for them to create a kind of a solid structure. And the cells here are not the same as the fibroblasts of ordinary connective tissues. They're reticular fibroblasts, and unusually for connective tissue cells, they actually attach to each other. There are gap junctions that appear at the ends of these processes. And when the cells make their matrix fibers, the fibers don't simply get exported to the surrounding region, but they remain attached to the cells. So type 3 collagen in reticular connective tissues remains attached to the cells that produce them. And I'll talk more about reticular connective tissues tomorrow. Okay, so now here is other collagens, collagen 5A, collagen 5A1, 5A2. Mutations in these collagens are part of a set of syndromes called Ehlers-Danlos Syndrome. And it was just descriptively designated by these two investigators. They didn't know what the molecules were. They didn't even know it was a collagen. They just looked at the symptomology. And one of the symptomologies is this very extensible skin. And other entities of Ehlers-Danlos have different kind of weaknesses and impairments in the body. So, for example, there are Ehlers-Danlos 4, 5, 6. Don't confuse this with the collagens. The syndromes were named before anybody knew about collagen. So Ehlers-Danlos syndromes 1 and 2 involve both collagen 5. So just keep the collagen separate. And I'm not asking you to memorize the syndromes by number or anything like that, but I just want to give you an impression of the disorders. Some of them involve just the amount of synthesis of a protein. So for example, Ehlers-Danlos 4 involves decrease in type 3 collagen. This could be due to something impaired about the promoter of type 3 collagen. It doesn't need to be the actual protein coding region of the gene, but maybe the enhancer or the promoter that makes the gene not expressed as efficiently. So, and you have symptomology, arterial, intestinal, and uterine rupture, thin, easily bruised skin. This entity decreased cross-linking of some collagens, probably type 1, skin and joint hyperextensibility, decreased hydroxylysine. Well, hydroxylysine, lysine is an amino acid that's prominent in collagen. It gets hydroxylated by an enzyme. So if that enzyme is mutated, you can have decreased hydroxylysine. So the gene for the collagen is fine, but the gene that processes, whose protein processes the collagen, is not fine. Okay. Here is the N-terminal propeptide is not removed. Here's a decrease in type 1 collagen, decreased hydroxyproline, okay, so on. But Ehlers-Danlos syndrome is not totally impairing. Here's a recent news item and a television program, Ninja Warriors, a woman competed who had Ehlers-Danlos syndrome. So don't know what the entity she had was, but it was prominently in the news that she was perfectly well equipped to compete in these athletic competitions and so on. Okay, now another kind of connective tissue is called elastic connective tissue. So here is elastic connective tissue. It has ordinary looking fibroblasts labeled F here, and it has ordinary looking type 1 collagen. Now here are some fibrils in longitudinal section with the stripes and in cross section here. But then you have these masses of material labeled E. That's elastin. Elastin can form fibers, or it can form sheets, or it can form kind of amorphous masses, and it stains like this. So what is elastin? Elastin is a protein that behaves like rubber. It is cross-linked, and if you extend it, it will want to snap back like a rubber band. Okay, now real rubbers are not proteins. They're terpenes and terpenoids, but this is a protein, but it behaves like rubber. So how can it behave like rubber? So you have these proteins that want to scrunch up. Their equilibrium configuration is kind of a folded structure like that. And they're cross-linked by disulfide bridges. And if you stretch a tissue that has elastin in it, the proteins want to refold. So it'll pull the tissue back. So why do you need a matrix component like this? What kind of tissues? So think about your aorta or any large artery. It gets distended when the blood is passing through, but then you want it to recover its shape very quickly, so it snaps back. The wall of the aorta is very rich in elastic connective tissue. Similarly, with the airways of the respiratory system, you have a lot of elastin there. It gets blown up with air, and then when you release the air, you want it to snap back. So that's, you have elastin. Then it's also in other places that don't, they're not like tubes, but like your external ear. It's, you could go ahead and do it. It's, it has elastic cartilage. It's cartilage with elastin, and it's rubbery. If you look at a rabbit's ear, it's not rubbery in the same way. It's just more stiff. But human ears are elastic cartilage. Now, elastin doesn't come just by itself. It has a scaffold protein called fibrillin. So, so here is elastin under the electron microscope, and here are microfibrils that are made of fibrillin. Fibrillin. And here's a schematic. So you have this elastic fiber with these microfibrils of fibrillin. And fibrillin is very important. Every place that you have elastin, you have fibrillin. And you also can have fibrillin by itself. The ligaments of the lens of the eye are very rich in fibrillin, but there's no elastin there. So fibrillin can function on its own, but mostly it functions with elastin, and elastin never functions on its own. And fibrillin has a well-known, well-characterized set of mutations that are collectively called Marfan syndrome, and they lead to certain body types and morphologies. So here is a family of people with Marfan syndrome. Note the long fingers, the extended body proportions of the face, and so on. This is not impairing cognitively or anything like that. In fact, people have speculated that Abraham Lincoln, you must have seen pictures of him, had a form of Marfan syndrome. There are mild forms and there are severe forms. The more severe forms lead to premature aortic rupture or impairment of the visual system because of the fibrillin's involvement there. And for a long time, it was thought that these mutations in fibrillin, Marfan syndrome, it was mainly structural. It was mainly the mechanical integrity of the connective tissues. But in fact, it goes beyond that. So like here are mesenchymal cells or fibroblasts, and they're sitting in a matrix. It's rich in fibronectin. And here are fibrillin microfibrils. Here's one with Marfan syndrome. So just all along these fibrillin microfibrils are attached growth factors. And the growth factors are in a latent form. They are present with extra pieces that make them not active as growth factors. In order for the growth factors to be active, and this is a very controlled thing, proteases come along and cleave the growth factors, BMP or TGF-beta, and these growth factors modulate the function of the connective tissue. They cause it to make more matrix or less matrix, but they're very important in the proportions of extracellular matrix and growth. So in these impaired microfibrils, somehow the growth factors that are attached to them in the pre-form are more susceptible to these proteases. So you have this greater abundance of these active growth factors in the matrix. It just deranges the tissue. The tissue is not well controlled if the fibrillin is mutated. Okay, so this kind of brings back this theme of these proteases that are so important. Here, it's not the proteases that are impaired, but it's the fibrillin that's impaired. But I just want to tell you a little bit about these metalloproteases. This is a figure from a paper on breast tumors. Cells are tumor cells, But the important thing here is that there are these MMPs, which are matrix metalloproteinases, which I showed you were important for collagen processing, for growth factor processing. And there are all these different cells associated with tumor growth: endothelial cells, myofibroblasts, the cancer cells themselves, the fibroblasts, the neutrophils, and so on. And all of them are making one or another matrix metalloproteinase, or TIMPs. TIMPs are tissue inhibitors of metalloproteinases. And they're extremely important too, because they keep the metalloproteinases in check. And you find that metalloproteinases and TIMPs are both involved anytime tissues are being remodeled. So during development in the embryo, these things are active, along with the production of matrix. Inflammation, they're active. Repair, they're active. Cancer, they're active. Anytime a tissue is undergoing change and remodeling and cells are relocating. Okay, so they're extremely important. Fibronectin, that term has come up several times. It's a protein that has two subunits attached by disulfide bridges. It has collagen binding domains, cell binding domains. Cell binding domains are the part of the protein that binds to integrins. Heparin binding domains binding to proteoglycans. And fibronectin is unusual in that, think about collagen. There are 27 different kinds of collagen, more than 27 different kinds of genes. It's only one fibronectin gene. But there are many different fibronectins throughout the body and the different tissues. And in fact, even within one fibronectin, there are the two chains are different. So how does that happen? It's alternative splicing. The messenger RNA gets made. It gets different pieces of it get excised and other pieces get joined up so that you have different protein domains, even though it's specified by the same gene. Okay, so you have more than 27 different kinds of fibronectin, but all specified by one gene. And of course, since we're diploid organisms, we have two copies of that gene. But apart from that, there's one fibronectin gene. About fibronectin in development. I'm just giving you one example here, but anytime you have tissue morphogenesis in the embryo, you have fibronectin involved. So this is the development of the limb, and the cells in the limb coalesce. They undergo something called condensation, and that leads to the formation of bones. First you get cartilage, and then the cartilage gets replaced by bone. But it's fibronectin that is important in mediating this clustering of the cells. And you can show that experimentally. So these are cells from, in this case, a chicken embryo, but they're put in a dish at high density, and they come from the developing limb, the limb bud. And the cells undergo condensation. And you can see the cells are more highly packed here and here and here. And if you do an immune stain, you find that those highly packed regions are very rich in fibronectin. And fibronectin is one of the molecules that promotes this aggregation. What Dr. Luria told you about lectins, well, there are some lectins that act even earlier than fibronectin, but fibronectin is extremely important as well. Okay. Now, I've talked about proteoglycans, and I've mentioned glycoproteins. These are two different species of hybrid molecule. A proteoglycan has a core protein, and it has O-linked glycans, these glycosaminoglycans that I told you about. Glycoproteins are mainly protein, and they have these small branched oligosaccharides. They're not straight chains, and they are usually the sugar arrangement is different. The sugars in these glycosaminoglycans are assembled from dimers, where you have two different sugars linked to each other, and then those are repetitively assembled one after the other. So these long straight chains have these repeating patterns. You don't have that in these short oligosaccharides. They have different functions. These are mainly structural. These are more they're kind of informational and signaling. So let's talk about the proteoglycans. You have proteoglycans that are integral membrane proteins. So here's the lipid bilayer of the plasma membrane. The core protein is stuck through the membrane, and you have the glycosaminoglycan chains, which are not always the same, kind of studded along the protein. This is called syndecan. This is glypican. This is attached by a kind of a lipid insertion into a phosphoinositol glyceride chain stuck into the plasma membrane. And then most proteoglycans are actually free in the extracellular matrix. They're not attached to the membrane at all, or maybe indirectly by, there'll be integrins that are attached to fibronectin, which in turn are attached non-covalently to the proteoglycan. And then this is just common glycosaminoglycans: hyaluronan, heparan sulfate, chondroitin sulfate, dermatan sulfate. And you can see that there are these disaccharides that are attached to each other, and then they're just repeated many times. You'll get this in biochemistry. This is just for illustrative purposes. Not going to be on the test. Okay, now I just want to point out some junctions. You've heard a lot about these junctional adhesions, tight junctions, adhesion belt. The adhesion belt are integral membrane proteins like cadherins that are attached to actin. You have desmosomes where you have proteins that are attached to intermediate filaments. And then you have gap junctions. Okay. Now you have counterparts to some of these at the base. So you have hemidesmosomes, which Dr. Luria told you about, attached to the basal lamina. You have focal contacts, which are the half counterpart of the adhesion belt. And this is attached to the actin cytoskeleton. These things tend to be more transient. The cells are moving and relocating with respect to each other. So they're kind of less stable. Then you have some non-junctional complexes. They're important in development and in the immune system. We won't get into that right now. But as far as the basal lamina is concerned, it's not only under epithelial cells, or not only at the epithelial connective tissue interface. So here's the basal lamina, the yellow, and the green is the reticular lamina. But you also have it around muscle cells, particularly like smooth muscle cells. Skeletal muscle cells will have a basal lamina. And in the kidney, you have this unusual situation where you have two epithelial cell types that are confronting each other at the filtration barrier. So you have the endothelial cells of the blood vessels, and then you have the epithelial cells of, they're called podocytes. And together they produce a very thick basal lamina that acts as a filtration barrier here. And you'll hear about that later on in the term in the kidney lecture. So back to this picture again. And what I want to point out here is that you have this complex structure with all sorts of, I'm showing you the collagens here, but if we go over here and we look at the laminin will be located over here, directly attached to the epithelial cell, and will mediate the connection to the type 4 collagen. And this is the structure of laminin, triple helix with a integrin binding domain, proteoglycan binding domains, collagen binding domains. And if you look at a two-dimensional rendition, and this is type 4 collagen, laminin, perlecan, integrin, you can see how complex the organization is and what things are binding to what other things. So this is the basal lamina is a very complex, well-organized structure. Okay, so here are anchoring junctions, which are the excuse me one second. These are the counterparts of the adhesion complexes, and they're attached to actin. And here is the basal lamina. So you have mechanical forces within the cell, and they're interacting with the extracellular environment. That's very important in morphogenesis and remodeling of tissues. The interplay between the mechanical properties of the matrix and the mechanical properties of the cytoplasm, the cell interior. Okay, and if you pull on the matrix, or if the matrix pulls on the cell will feel it and change its biosynthetic properties, and correspondingly, the cell can put out more extracellular materials, which change the composition of the matrix and change its mechanical properties. So you've heard about integrins. Integrins are dimers. There's an alpha chain and a beta chain. The alpha chain gets clipped, and then it gets linked by disulfide link. Who knows why? I've never seen any rationale for that, but that happens. You have an internal region which binds to talin and binds in turn to things like intermediate filaments, either keratin, vimentin, desmin, and so on, or it can bind to actin. And then you have an extracellular region which binds to fibronectin or laminin or directly to collagens. And so here's an extracellular matrix protein binding to the integrin. And all of these components, these adapter proteins inside, are subject to modifications like phosphorylation particularly. And that changes the affinity of these proteins for one another, and it can either enhance the linkage to the cytoskeleton or relieve the linkage to the cytoskeleton. So as the cell is moving, it can grip the linkage to the extracellular environment, or it can let go of it. So there are things that go on inside the cell that change the, if this gripping down here is altered, it could let go of the extracellular, change the conformation of the integrin. So you have what's called inside-out signaling, and you also have outside-in signaling, because you can have changes in the matrix. Maybe a metalloproteinase will become active, will break down some of the matrix, and then cause this to disconnect, and then that changes what goes on inside the cell. So I think Dr. Urias showed this picture. So you have this complex extracellular matrix. You have integrins that are linked to the cytoskeleton. And very importantly, and more realized in the last couple of years, the mechanical effects of these matrix linkages on the nucleus actually changes gene expression. So it used to be thought that gene expression was controlled simply by soluble signaling molecules that get into the nucleus and change the activity of the transcription factors and so on. But now we know that just tugging on the nucleus, changing the shape of the nucleus slightly, can change the organization of the chromatin, the chromosomal material, and alter the rate of expression of certain genes. So there's not only chemical signaling, chemical signal transduction, but there's mechanical or mechanotransduction of gene expression. Okay, I think that's it. Any questions?